जैसे कि आपको मालूम होगा कि हमने हाइब्रिडाइजेशन, प्रोब हाइब्रिडाइजेशन, ऑटो रेडियोग्राफी तक बात कर चुके हैं रिगार्डिंग सदर्न ब्लॉटिंग. दैट वाज द लास्ट स्टेप ऑफ सदर्न ब्लॉटिंग. अब समटाइम्स क्या होता है कि हमें एक प्रोब के बाद दूसरे प्रोब को हाइब्रिडाइज करने की ज़रूरत पेश आती है, तीसरे प्रोब को हाइब्रिडाइज करने की ज़रूरत पेश आती है. तो इसके लिए हमारे पास ऑप्शन मौजूद है कि हम सेम मेम्ब्रेन को यूज़ कर सकें, बजाय इसके कि हम 0 पॉइंट से दोबारा से स्टार्ट करें कि डीएनए एक्सट्रैक्ट करें, इलेक्ट्रोफोरेसिस करें, मेम्ब्रेन पे इमोबिलाइज करें एंड देन वी प्रोसीड फर्दर फॉर सदर्न हाइब्रिडाइजेशन. This is possible to strip the probes from the membranes and use the same membrane for reprobing. In many cases, this is possible. This is possible to remove the probe and reprobe the membrane to get another result from the same membrane. लेकिन इससे एसोसिएटेड कुछ मसाइल हैं, कुछ प्रॉब्लम्स आते हैं जिसकी वजह से we cannot repeat the process for number of times. Keeping in view these problems, we have to decide how many times nylon membrane can be reprobed or how many times a nitrocellulose membrane can be reprobed. Irreversible binding is one of the problem. Sometimes the negligence causes the irreversible binding of the probe to the टेम्पलेट डीएनए टू दी सैंपल डीएनए. और ये उस वक्त होता है जब ड्यूरिंग सदर्न ब्लॉटिंग हम मेम्ब्रेन को ड्राई कर देते हैं फॉर लेंथी पीरियड ऑफ टाइम. इफ द मेम्ब्रेन बिकम्स ड्राइड, द प्रोब बिकम्स इरिवर्सिबली बाउंड विद द डीएनए सैंपल एंड कैन नॉट बी separated, cannot be stripped. Then fragility of the membrane and leaching of the nucleic acid with every cycle of probing is going to be the next problem. In order, if you want to reprobe the same membrane with more than one probes, so ensure it that the membrane don't get dry during the process of southern blotting. In that case, you can use that membrane again and again for reprobing after stripping of the previously used probe. Nylon membranes can be used 5 to 10 times depending upon your handling and depending upon how many harsh washing steps are used to wash the probes. Nitrocellulose membranes, they are quite fragile. They cannot sustain or they cannot withstand this washing process more than 2 to 3 reprobing cycles. So they can only be used for 2 to 3 times for hybridization. And when we talk about reprobing, keep in mind we have to go through number of washing steps. When we go through with the number of washing steps, partially leaching of the nucleic acid keeps on going. Ultimately, the results don't reflect the exact amount of DNA immobilized on the membrane. So we have to be careful when we are working with the DNA sample and the probing that if the signals are not coming, it doesn't mean that the DNA is not present. There is likely possibility that the DNA has leached out to such an extent that the probing is there, but the signal is not quite strong that could be recorded.